Hey everyone, and welcome. Have you ever wondered how a simple cut on your finger heals, or, how you grew from a single tiny cell into the person you are today? Well, the answer to all of that lies in the cell cycle, one of the most fundamental processes in all of biology. So in this explainer, we're going to break it all down step by step just for you. So here's the game plan. We're going to start with the big picture of a cell's life. Then we'll zoom right in on the two main types of division, mitosis and meiosis. After we compare them, we'll take a look at what happens when this whole process goes wrong and why that is so important for our health. Okay, let's kick things off with the big picture, the life story of a single cell. And this story, it applies to pretty much everything alive, from the simplest little bacteria all the way up to us, because every living thing is a product of cells growing and dividing. So what exactly is the cell cycle? Well, you can think of it as this incredibly precise sequence of events. It's like the cell's internal software that tells it exactly when and how to copy its genetic material, its genome, and all its other parts, and then, finally, to split into two. This is literally how we grow and how we heal. The entire cycle really boils down to two major stages. First up is a long preparation period called interphase. And get this, the cell spends a whopping 95% of its time here. All of that is followed by a much shorter, much more dramatic period of actual division, which we call the M phase. That's M for either mitotic or meiotic phase. So let's dive into that first part, interphase, and see how a cell gets ready for the big split. And let me be clear, this is not a resting phase at all. It's actually a period of intense activity and growth. A great way to think about interphase is like you're preparing for a big move. In the first stage, G1 or gap 1, the cell grows, it gets bigger, it gathers resources. Then comes the most critical step, the S phase, S for synthesis. This is where it makes a complete copy of all its most important furniture, the DNA. And finally, in G2 or gap 2, it does some last-minute packing and final prep before the big move, or in this case, the big split. Okay, listen up, because this is a super important point that trips up a lot of students on exams. In the S phase, the amount of DNA in the cell doubles. It goes from what we call 2C to 4C. But, and this is key, the chromosome number, 2N, stays exactly the same. So why is that? Well, it's because we count chromosomes by their centromeres, that little part that holds them together. And even after the DNA is duplicated, as long as the two copies are attached, it's still counted as one single chromosome. And this picture right here, it shows you exactly what's going on. We start with a homologous pair, one chromosome from mom, one from dad. During that S phase, each one gets copied perfectly. This creates that classic X shape that you always see in textbooks, where the two identical halves, which we call sister chromatids, are joined together at that central point, the centromere. All right, with all that prep work done, we're ready for the main event, or at least the first main event, mitosis. This is also known as equational division, and that's because the two new cells that are made end up with an equal and totally identical set of chromosomes as the parent cell. Mitosis is kind of like a 4-act play. And to remember the order for your exam, just use the awesome mnemonic PMAT. That's P for prophase, where the chromosomes pack up, M for metaphase, where they meet in the middle, A for anaphase, where they move apart, and T for telophase, where you get two new cells at the end. PMAT. Easy, right? So let's zoom in a bit. In prophase, that messy DNA starts to coil up super tight into those visible X-shaped chromosomes. Then, in metaphase, a network of protein fibers, kind of like little ropes, forms what we call the spindle. These fibers grab onto each chromosome at a special little handle called the kinetochore, and they pull them all into a perfect line right down the middle of the cell, the equator. And then the real action starts. In anaphase, the centromeres holding those sister chromatids together just split, and poof, the spindle fibers yank the identical copies apart toward opposite ends of the cell. Now, once they're separated, they're considered individual chromosomes. Finally, in telophase, the chromosomes arrive at the cols. A new nuclear membrane forms around each set, and the chromosomes start to relax and uncoil again. Okay, let's switch gears completely. Now we're going to talk about the other type of division, meiosis. This is the special process that's used for sexual reproduction, and its whole purpose is to create genetic diversity. So unlike mitosis, the goal here is not to make clones. Meiosis is what we call a reduction division, and that just means it cuts the number of chromosomes in half. It starts with one diploid cell, di means two, so it has two sets of chromosomes, and it ends with 4 haploid cells. Haplo means single. like sperm or eggs, each with only a single set of chromosomes. Now, meiosis has a couple of really unique tricks up its sleeve. First off, it's a two-part process, meiosis 1 and meiosis 2. But the second thing, this is the real game changer, something called crossing over. During prophase 1, the homologous chromosomes pair up and literally exchange bits and pieces of their genetic material. It's like they're trading arms and legs. This genetic shuffling creates brand new combinations of genes, and it's the fundamental reason why you and your siblings aren't identical clones. Okay, here's another pro tip for your exam. That second round, meiosis 2, it actually looks a lot like mitosis. The chromosomes line up in the middle, the sister chromatids get pulled apart. It's all very familiar. The big difference is that the cells going into meiosis 2 are already haploid. They already have half the number of chromosomes to begin with. So now that we've walked through both processes, let's put them head to head. Honestly, seeing them side by side like this is the absolute best way to make the differences stick in your brain. Seriously, screenshot this slide. It's your ultimate cheat sheet. The easiest way to remember all this is just to focus on the goal. The goal of mitosis is to make identical copies for growth and repair, so it does one simple division. The goal of meiosis is to create genetic variety for reproduction, so it needs 2 divisions and that super special crossing over step to shuffle the genes. The result? 2 identical cells from mitosis versus 4 unique cells from meiosis. So you might be thinking, okay, that's a lot of biology, but why does it matter so much? Well, let's talk about why controlling this whole process is absolutely critical for our health. So, how does the cell keep all this in check? Well, it has these internal police officers, or quality control stations, called checkpoints. There are a few of them, but the major ones are at the end of G1, right before M phase, and during metaphase. These checkpoints scan for problems, like DNA damage, or maybe the spindle fibers didn't attach correctly, and they can hit the brakes on the whole cycle if something's wrong. And this, right here, is the critical link. Cancer, at its very core, is a disease of the cell cycle. When these checkpoints fail, when a cell starts ignoring the stop signs, it can begin to divide uncontrollably over and over, and that is what leads to the formation of a tumor. All right, let's do a super quick, rapid-fire review for your exam. If you remember nothing else, remember these points. The cell cycle has 2 parts, interphase and M phase. Mitosis makes 2 identical body cells. Meiosis makes 4 unique sex cells. Crossing over is the key to genetic variety. And checkpoints are what keep the whole process from going off the rails. Got it? And I want to leave you with one last thing to chew on. Some of our most important cells, like the mature neurons in your brain, they actually exit the cycle permanently. They go into a state called G0 or GO. They never ever divide again. So why do you think it's so critical for a complex organism to have cells that don't divide? Pondering that question really reveals the true elegance and importance of cell cycle control.